Good morning, students. In this class, we'll be discussing about nitrogen metabolism. As you all know, nitrogen metabolism is a physiological process and involves a series of biochemical reactions. And here, for the nitrogen metabolism, there must be a constant supply of nitrogen to the plants and other organisms. Also, there should be a steady state of this nitrogen in the atmosphere also. So at the same time, nitrogen should be supplied to the organisms and it should be maintained in a constant level in the atmosphere also. So a wide range of reactions, a series of reactions are taking place to maintain these levels. And there are different pools of nitrogen, and these pools are atmospheric pool. Atmosphere acts as the source of nitrogen. Also the second one is soil and groundwater pool, and the third one is biomass pool. So this exchange of nitrogen in between these pools is known as nitrogen cycle. And nitrogen cycle has some major reactions, that is nitrogen fixation, denitrification, ammonification, nitrification, and nitrate assimilation. First, what is ammonification? Ammonification, it is the conversion of organic nitrogen into ammonia. So the formation of ammonia from organic nitrogen, organic matters is known as ammonification, and this process is made happen with the help of certain organisms like bacteria, fungi, and certain of actinomycetes. So these type of decomposing organisms, they convert the organic matter, the organic nitrogen, into ammonia. Okay, the nitrogen present in the organic matter is converted to ammonia, and the certain amount of energy is released in this process, and this released energy is used by the bacteria for their metabolic purposes. The second type of reaction is nitrification. Nitrification is the process wherein oxidation of ammonia to form soil nitrate, and two steps are here with the help of two bacteria commonly known as nitrifying bacteria. The first step is oxidation of ammonia to nitrate by nitrite bacteria, and nitrite bacteria's example for nitrite bacteria is Nitrosomonas is one example, and another example is Nitrococcus bacteria. And in the second step nitrite is further oxidized to nitrate, and this is with the help of nitrate bacteria. Example is Nitrobacter. Okay, so we have gone through two major steps, ammonification and nitrification. Denitrification is the process where the reduction of nitrate to nitrogen, molecular nitrogen, is taking place, and here the bacteria is denitrifying bacteria. Okay, so in case of nitrogen fixation is the reductive conversion of atmospheric nitrogen to nitrogenous compounds, and this process, it can be biological nitrogen fixation or non-biological nitrogen fixation. In case of non-biological nitrogen fixation, it is a physicochemical or electrochemical conversion, and it may happen with the help of lightning, radiation, thunderstorms, etc. And biological nitrogen fixation is carried out with the help of some living organisms called nitrogen fixers, and it may be prokaryotic, and nitrogen fixers may be bacteria, fungi, lichens. All these can perform nitrogen fixation, and they can be divided into two groups: free living and symbiotic. Free living nitrogen fixers include some cyanobacteria, and a wide variety of bacteria are there, which may be freshwater or marine. The second category is symbiotic nitrogen fixers, and this symbiotic nitrogen fixers, they are inhabiting within other organisms and performing the nitrogen fixation. They may be lichens. Lichens are a symbiotic nitrogen fixing organisms which inhabit the thallus of certain algae, and coralloid roots of cycas, they are also having a symbiotic association, and likewise several other examples are there. So nitrogen fixation can be of two types: biological and non-biological. And in case of biological nitrogen fixation, it has to form root nodules. For symbiotic nitrogen fixation, there are two strains of rhizobium. Okay, one is slow growing, another one is fast growing. And these rhizobium, they form a nodule in the roots. So the root nodule formation is a complex process wherein the bacteria and the root interact with each other, and there is specificity in this association, and this specificity, it is determined by a protein of the host cell called lectins. So this lectins, it selectively interact with the proteins in the bacterial capsule, and thus they determine the specificity of host and the symbiont specificity. So what happens is, the lectins, which are the key molecules in determining the specificity of the interaction, and calcium is also having a role in enhancing the ability of the roots to accommodate these rhizobium within it, and the formation of root nodules begins with the infection in the root hairs. The free living rhizobia, they infect the root hairs, and the presence of these roots within the vicinity of these bacteria, they enhance the bacteria. They come and colonize near the rhizosphere, and during this, the root also synthesizes and releases tryptophan to the soil, and the bacteria, they absorb and metabolize this tryptophan to produce auxin. IAA is produced, which is a auxin type, and this will promote the growth of the roots. This rhizobium is attracted towards the host, particularly the root hairs, and this is an example of positive chemotactic movement. And Once this rhizobia, they have colonized within the rhizosphere, they synthesize some signal molecules, and these signal molecules may be the root hair curling factors and the nodulation factors, which can be also known as node factors. And root curling factors, they cause the curling and curling movements of these roots, and the other one, the node factors, they cause the increased production of root hairs, and shorter and thicker roots are developed with this effect. This rhizobia they also release some signal molecules, some mitogenic signal molecules, which causes or which stimulates the cell division in localized regions, particularly within the root cortex, to form the primary nodule meristem. And later it eventually also develops the secondary nodule center, and this is formed within the pericycle. And after some time, this primary and secondary nodule centers, they fuse with each other and form a complete nodule. And in the next stage, what happens is rhizobia, it penetrate the host cell wall and get into the cells through the plasma membrane. And since they cannot digest cellulose, they penetrate the tip of root hairs where cellulose is absent. The root hairs, it ceases its growth and the plasma membrane undergoes invagination, and within this in vesicular form or within this in invaginated vesicle like structure, these threads of infection threads will be seen, and these bacteria, they invade this space, and it elongates and they multiply within this space, and they degrade the cell wall of the cells next to them, and thus causes the elongation of infection tubes. And shortly, these bacteria, they stop dividing and enlarge in size, and they differentiate to nitrogen fixing cells, and these are called bacterioids. And the membrane which encloses these bacterioids is known as peribacterioid membrane. This nodule, they also contain a pigment, a pink colored pigment, which is known as leg hemoglobin, and this is oxidized to form brown color. And the development of leg hemoglobin, it is a very important step for the nitrogen fixation process. And the biological nitrogen fixation, it is enzymatic, and it occurs as the same way as we discussed in the initial stages of this class. And two enzymes, nitrogenase and hydrogenase, are there. Nitrogenase is also known as dinitrogenase, and the hydrogen donor system is the protons and electrons from this are acting as the donors in this reaction. And a major hydrogen donor is the NADPH. First stable compound in this reaction is the ammonia, and nitrogen fixation, it leads to the reduction of this dinitrogen molecule to form two molecules of ammonia. And whole process is explained here in this figure. this is a plant and the soil rhizobium, and this nitrogen is present in the atmosphere, and this atmospheric nitrogen is fixed by the nitrogen fixing bacteria. Ammonification is this process wherein the bacteria, ammonifying bacteria, converts the nitrogen in the organic matter to ammonia. Also the nitrogen fixing bacteria, they also convert the atmospheric nitrogen to ammonia. This ammonia is reduced to ammonium with the addition of one more proton, and this ammonium is converted to nitrate by nitrifying bacteria. Nitrate is taken into the roots, and here they are converted to ammonium and further to nitrogenous compounds and exported to different parts of the plant. Okay, here you can see the formation of root nodules. Root hairs are there, and in the vicinity of root hairs certain rhizobiums colonize, and they get into the root hairs. They colonize within the cortical region and form bacteroids, and there they multiply in number, and after some time they stop their multiplication and they form a vesicle like bacteroid structure, and this is the bacteroids. Okay, so this is the process of nitrogen fixation. And microbial nitrogen fixation, that I have already posted a video regarding that portion. So hope you all understood. Thank you.